Hey, this is Roy with covering questions 1 and 2 today for the group problems. Question 1 was pretty straightforward. It's just straight from the reading. It talks about, or pretty much, it asks to indicate the group of the enzymes that covalently link amino acids to tRNA molecules. It stated in the reading that it was aminoacyl tRNA synthetase, and then it asks the indicate the next, or indicate 4 proteins that consume GTP during translation. And these were the IF3, IF2, EFTU, and EFTG, and those were all during the I believe the elongation and initiation steps. Then it continue on, indicate 1 molecule that functions as a ribozyme during translation, and that's the peptidyltransferase. D was indicate 2 proteins that recognize stop codons. I thought this was really interesting because RF1 recognizes 2 different stop codons, and then RF2 recognizes the other 2 stop codons. So I was like, oh, that's pretty sweet. I didn't know that there's 2 different proteins. E is indicate 1 molecule that forms hydrogen bonds with the Shrine-Delgado sequence of the mRNA, and that is the 16S subunit of the 30S ribosomal subunit. So that's a unit of a unit for that. So that's question 1. It's pretty straightforward. And then I also had question 2. The first amino acid in prokaryotic proteins is the FMET. In eukaryotic, it's just MET. And then an E. coli protein called EFTU delivers charged tRNAs to the A site of the ribosome during translation and elongation. The initiator tRNA binds to the P site of the ribosome, and then all the remaining charged tRNAs bind to the A site, and that's the A site's where they get bound together to make proteins. D is the EF-G is a translation factor that helps ribosome move 1 codon from the 3 direction along the mRNA. So just kind of that's the factor, and I think that's one of them uses GTP2. The IF3 is a translation factor that guides mRNA to the 30S ribosome subunit. It's the that's just in the reading. And then the AUG codon is a start codon. That's the only start codon there is, the AUG. It codes for F-methionine. And then the UAA is an example of a stop codon. In the slides, it says UAA, UAG, and UGA are all 3 of those are stop codons. And oddly enough, it talks about how they're read by the RF1 and RF2 proteins. Degenerate is the term that indicates more than 1 codon can specify for the same amino acids. And then the final amino acid located in the C terminus of the polypeptide. That's just protein synthesis there. So those are my problems. That's problem number 1 and problem number 2. I will let, I think it's Ashley take number question 3.
Okay, so I have problem 3. This one is asking to list all the possible sequences for different mRNA codons. For letter A, it lists it in the 5' to 3', so it's UUG. For the anticodon, it'll be 3' to 5'. 2 different possibilities. We have AAC and AAU. The ones that I have underlined and bolded, those are the ones that change. The first 2 will always stay the same. So for the second one, letter B, we have AGU. So that can translate to UCA, UCI, or UCG. I plays a part in this because it can attach to a codon that either has U, A, or C on the 3' end of the codon. For letter C, you have UAC, and that can translate to AUG or AUI. For letter D, you have AUU, that translates to UAA, UAG, or UAI. For the last one, you have GCA, and that'll translate to CGU or CGI. And that's it for problem 5, so I'll turn the time over to Kyle.
Hi guys, so I'm going to be doing problem 4. We had a group member who didn't show up, so this is kind of last minute, but I already had it done. So in question A, it's asking, so it says that you have a protein X, that weighs 218 kilodaltons, and each amino acid in that protein weighs, on average, 125 daltons. And so I used a picket fence method. I don't know if you'll be able to see it. Oh, it's backwards too. Anyways, so I took the kilodaltons divided by how many daltons there are per one amino acid, and I got 1,744 amino acids. And number B, they want to know how long this amino acid chain protein X is. And it didn't really tell you what to do it in. And so I have two different answers here, and hopefully one of them is right. So if there's 1,744 amino acids, there are 3 base pairs for that one acid. And so I times that number by 3 and then added 3 for the N codon that wouldn't have been transcribed. And I got 5,235 base pairs. And then I also calculate that into nanometers on the basis that each base pair is 0.34 nanometers apart. And I got 1779.9 nanometers would be the length for that protein X. For part C, they want to know how many GTP are consumed in creating this protein X. And so there are 4 GTPs that are consumed per amino acid on the protein X. And so when you times 4 by 1744, you get 6,977 GTPs. And for part D, they want to know how many ATPs were used to create this protein X. And there is 1 ATP used per amino acid in the chain, and that would be 1,744 ATPs used. And the answer to question 4E is the amino acyl tRNA synthetase. And that is everything for question 4. And now I'll be going on to question 5. All right, I'm almost there, sorry. All right, so they gave us a big list of all of these different factors, and they wanted to know which one are in translation, elongation, termination, part of the 70S ribosome, and found in mRNA. So the ones that are involved in translation include the 16S RNA, the Shine-Dalgonaro sequence, the start codon, IF3, IF2, and tRNA FMET. And for question B, which is involved in translation elongation, is the 16S rRNA, EFTU, EFG, 23S rRNA, and lysyl tRNA synthetase. And that last one, it is the same thing as the amino acyl tRNA synthetase. Each amino acid that gets incorporated in a chain, they each have their own synthetase, and so the amino acyl rTNase is just kind of like the broad name, and the specifics include part of the amino acid in the beginning of their name. For 5C, which of the above is involved in translation termination? And that's recognition factor 1 and UGA, which is a stop codon. For 5D, which of the above is component of the 70S ribosome? And that would be the 16S RNA, which is part of the 30S subunit, and then the 23S RNA, which is part of the 50S subunit, and the 30 and the 50S make up the 70S ribosome. For E, which of the above is found in the mRNA? So that would be the Shine-Denaro sequence, which helps with initiation, and the end codon and the start codon. All right, that's all we have for you guys today. Hope you enjoyed it. See you later.